I think we'll still let a few people filter in. So let's do a quick temperature check. Is everyone here for the non-programmers piece who's not actually doing the lab, like the cool people on the cool side? Yeah? Everybody here is cool? Yeah? Okay, good. We just want to make sure, because the CIDA lab is actually right next door. So if that's your track, you can go right next door. Really quickly, just want to sort of lay out the plan for this afternoon. Like I mentioned, the CIDA lab is next door. On this side, what we're going to do is pretty much go over and review the reports of the CIDA tool and how they can create, what they can create after running the incidence rates and propensity score analysis. In order to show us that, we have Dr. Noelle Corocos, and I'm going to give her a quick introduction to her. She has a background in infectious disease epidemiology and public health surveillance. She's been at the Department of Population Medicine at Harvard Pilgrim for 5 years, and she's worked on Sentinel since she's been there. She also works on an EHR-based distributed data network for chronic disease surveillance and infectious disease that reports for state health departments. Noelle's current Sentinel and Sentinel leverage work includes the IMPACT AFib clinical trial, a set of activities with FDA that we work with the Office of Counterterrorism and Emerging Threats that focuses on influenza-related analyses, and the lymphoma HOI chart validation project. So we're going to go through that, and then afterwards we're going to also introduce Jane to kind of follow up on the next piece. Thanks.
All right, good afternoon. All right, so I have the challenge of the after lunch session, so I'll do my best to keep it as lively as possible. So what I'm going to be talking about, all right, so I'm going to be doing the CIDA report interpretation. So as Anissa said, Judy's next door, walking people through how you go from designing your analysis to creating the package itself. And what I'm going to do is show you what we see as epidemiologists in the operations center, as well as at FDA. So I don't run the package. That's why I'm not in that room. And so I'm going to show you what I see as one of the investigators at the operations center. Before I do that, I'm just going to do a quick overview of a couple things. Judy essentially gave you truly like a month's worth of training in a morning. So it was quite a crash course. And so I'm just going to go over in the highest level a few of the key things to keep in mind, and hopefully it'll help it sink in a bit. So this is the sort of high-level display of the different analyses, the different levels of analyses that we can conduct within Sentinel. We use, as Judy said, these SAS programs that we call tools that can be parameterized and can be used to ask a number of different types of questions and provide standard output. And that output is what I'm going to be showing you momentarily. This is the other view that Judy was showing you this morning, and this tells you about the different types of analyses within those different levels. And so I won't go into detail because Judy did that already, but I'll show you where we're going to be in a moment. So the agenda for the next half hour or so is really focusing on interpretation of the report contents. And I'm going to go through 3 different specific sets of reports following on what Judy already showed you. And so this is where we're going to start, is with the data that you'll get back for doing this type 2 analysis where you're calculating incidence rates, and follow-up time is being taken into account as well. So as a reminder, this is the query that's Judy set up this morning. So the question that FDA is interested in this example is looking at stroke following antipsychotic use. She set up, using that new query builder capability, medical utilization analysis. And then she set up the design diagram and query specifications for calculating incidence rates and doing a propensity score matched analysis, and how you really go through, or how we go through in collaboration with the FDA, identifying all the details of that kind of analysis. And this is what it translated into. So these are all of the things that Judy walked through. I'm not going to go through them now, but at the highest level, we're going to be looking at synthetic data based on the Medicare. The underlying data source was Medicare, but it was transferred into a synthetic data set We're looking at adults 18 to 65. They do not have dementia in their claims history that we can identify based on the different parameters we set up. We're looking at incident use of typical and atypical antipsychotics, and then we're looking for incident stroke, and we're defining that two ways, with intracranial bleed or ischemic stroke. And so that's the analysis that I'm going to walk through. And so getting right into it. So this next set of data that I'm going to show you, this is what we call our baseline table, covariate table. Think of it like a classic table 1 in a manuscript. So you want to basically get information on whichever cohorts you're interested in. The default output for this table is going to give you information on your cohort of interest by age, sex, race, year of exposure, so that's your index exposure, any user-defined conditions that you may have specified, medical and drug utilization metrics. So the slide that you'll see on this actually doesn't show you those metrics, but I'll describe to you what's available. There's also a comorbidity score. And as Judy pointed out, for each of the conditions that you're interested in, so you want to know among this population, among this cohort, how many people had history of diabetes, or how many people had history of acute myocardial infarction, you can define different windows of time to look for those conditions of interest. And all of that gets reported in one table. So this is part of one of those tables. And what you'll get back for the query that was set up this morning is you'll get one of these for your atypical antipsychotic users with respect to stroke and with respect to intracranial bleed. Same thing for atypical. So you'd get 4 table 1s. You're not seeing here the stroke outcomes, but you've washed out anyone with those conditions of interest because you're looking for incident use. So you'll see slightly different numbers. So again, you'd get 4 different cohorts created, and you'd get 1 table for each. And this is going to give you a lot of information to design your study. When Jane presents next, she's going to be showing you the data that the analysis that was the set of analyses that were conducted using real Sentinel data, and basically how each level of analysis was used to build onto the next, and how you can use this data to really inform choices that you make downstream. So what you can see here, let's see how this pointer does. So you've got up here your total number of episodes in patients. So they're identical numbers here. So this is that example where Judy was explaining you can choose to say, allow someone to enter the cohort once and take their first incident or index episode of interest, or allow them to contribute multiple that meet the eligibility criteria. In this case, you said just take the first, and so that's why the number of episodes and the number of patients is identical. In other cases, you would see more episodes than there are patients. Then you get information, obviously, on the age of the patients. This is that age at the time of the index exposure. You get the breakout by sex. You get the breakout by year of when those index dispensings occurred, and you can see the distribution, again, reflective of the synthetic data and what data was available at the time. And then you get some of the meat of the output, which is this recorded history of different conditions. And so these are all user specified. So you have to decide what are the conditions of interest, what are the codes that you want, and then what is the window that you want to look for these different conditions. In this case, the query was specified to look back 6 months for evidence of a code that was used to define one of these conditions. You can actually look back or forward, and it really depends on your question at hand, how you're going to use this covariate information. In this case, it was just looking back in time. And you can see here the proportion of the people in this cohort of typical antipsychotic users who have these different conditions. Again, I guess to really get the message on, these are synthetic data, so I wouldn't focus too heavily on the actual results, but instead to just see the type of output that you'll get. So this is your table 1. You'll get this for each cohort. You can get this kind of output with any of those different types of analyses within CIDA. So you can run a type 1 analysis where you're just calculating some background rates and you're not calculating follow-up time, and still get one of these. So it's this sort of add-on option that you can use for different kinds of analyses. All right, so now we're going to get right into what we're calling the type 2 report. So this is the report that is generated from doing that type 2 analysis where we're calculating incidence rates. And I'll walk through each of the columns and just give you some highlights. So just so that you can understand how to interpret some of the output. What's here is all of the columns. Sometimes, depending on what your analysis is, they may not all be relevant. And so sometimes we actually just hide the columns or remove them if they're not useful or not helpful to what we're doing. But here, the orientation is that you've got it's categorized by the outcome. So you've got ischemic stroke and intracranial bleed here. And then you've got your two different exposure groups. So you've got typical and atypical antipsychotic users, and same here. And they each get a row. And I'll just be going through each of the columns. So like you saw before, right, there's 24,720 typical antipsychotic users. There's the same number of episodes for them. You can see here the difference in the atypical versus typical users in terms of counts, and then you can follow through the numbers again. You're seeing a difference in the number of typical users here compared to the number here because of that washout of the outcome, right? We said you couldn't have in your recent history any evidence of one of the outcomes of interest, and so there's a difference that's generated as a result of that. So nothing terribly exciting here. That's just your counts. As we move on, this is now the eligible members. So this is your denominators, and there's actually sort of an important thing to keep in mind here, depending on the kind of analysis you're doing. So the strict interpretation or definition of what this is in the footnote, right? So it tells you who eligible members are. They have, on at least one day during your query period, met all of the different cohort criteria that you laid out. So in this case, they were 18 to 65 on at least one day. They met the enrollment criteria. They did not have evidence of dementia in a certain set of time, et cetera. So on at least one day, if I met all of those criteria, I would contribute one eligible member to that column. If I met those criteria for 2 years, I would contribute that number to the number of member days. Relatively straightforward. So this is the example here. It shows you all of the different criteria that were used to define who's in your denominator. It's pretty straightforward. It can get a little bit, there's the nuance of it is important depending on how you're using the tool. So again, here it makes sense. But there are examples where that eligible member count can be a little bit funny. And so we use this tool, and this will also give you a little perspective on how else you might use some of these analytic tools. So one of the projects that we're working on with FDA is a lymphoma health outcome of interest validation project. So we say, this is an electronic algorithm for lymphoma. Now we're going to identify people who have lymphoma based on that algorithm, and then sample them and look at their charts and validate it. So we use CIDA to do that first part, to actually pull the cohort. And so you can see here the algorithm that is proposed for the study, the details here. The person would have to have 2 lymphoma diagnosis codes within 6 months of each other. The first one is incident. And then they had to have a biopsy and imaging procedure codes in their history. So in other words, if you meet all these criteria, we're going to say, looks like you have lymphoma. We're going to sample from you and review charts. So we use CIDA to do that first part. So when we got the results back, these are all the different criteria that go into account for pulling that cohort. So we get the results back, and before we say to the sites that are participating in the study, go forth and find the charts for these people, I review the reports, make sure nothing looks odd, and I saw that the eligible members made no sense. I mean, the numbers were so small, and it took me maybe 10 minutes before I remembered what an eligible member is. And in that case, it's someone who meets these criteria, all of these criteria. And so when you add in these strict requirements for certain kinds of procedures or conditions that are rare, if you require that for cohort entry, your eligible members is affected accordingly. and so your denominator might be small. So it's just something to keep in mind. Despite the fact that I've worked in the system for years, it took me like a few minutes before I could understand why the data looked so odd. So the point here is that your eligible members represent your denominator. They met all the inclusion criteria for the cohort. And you contribute if you meet all those criteria for at least one day. So now moving down the column. So now I'll talk about the next set of output, which is your days at risk and years at risk. And so this is really the time that someone is at risk for the outcome. And you might remember the way the query was set up was look for, the number of days that I was dispensed the drug, allow 30-day gap, and then tack on 30 days at the end, because you think that someone is at risk for stroke when they're on one of these drugs while they're on it and for 30 days after. So that days at risk here is a summation of your dispensing data, so the actual data in your claim, plus those additional parameters. And so in this case, for most of it, for most people are contributing 60 days here, again, because of the synthetic nature of the data. And then years at risk is based on that. What you can see is that it really becomes clear when you look at days supply. So these two columns, this is like straight from the claims data. So I was dispensed a 30-day supply. I'm going to contribute 30 days here. And then you tack on your 30-day extension, and it gets summed up here for the days at risk. And then amount supplied is a reflection of the actual units that you were dispensed. So if I was dispensed 2 pills a day for 30 days, I'd be contributing 60 amounts there. So those are the different parameters in that case. So those are all of your at-risk time and some information on days supplied and amount supplied. So moving along, so it's out of order in terms of left to right, but adjusted dispensings and raw dispensing. So this is getting at a little bit of what Judy touched on this morning, that stockpiling parameter. So you can decide how you want to treat people who were dispensed the same drug multiple times on one day. So I think Judy talked about this a little bit this morning, that in this case, it's not particularly interesting. Not too many people in this synthetic data got 2 dispensings of one of the antipsychotics on the same day. And so even though that stockpiling parameter was turned on, it doesn't really do much. You can see almost identical counts. And I'm going to walk through what this means in a minute. But this is your raw data. This is the actual count of the number of dispensings. So if I got 2 dispensings on one day, and then 1, 6 months later, and you counted all of them, I'd contribute 3 here. The adjusted is taking into account the stockpiling and saying the 2 that I got on one day, don't treat them as 2, treat them as 1, because we're going to assume that you've stockpiled them and you're not actually taking them on the same day. So again, in this case, it's in this query and in this data set, it's not particularly interesting or exciting. I don't know if it's ever necessarily that exciting, but you're not seeing a big difference. This is just an illustration of how that works. So this is someone who is sort of the perfect patient. They get a 30-day supply, and they go and pick up their dispensing exactly on time. And they would show up in those 2 columns of data as contributing 3 dispensings from the raw data, and then the adjusted would also be 3, because they didn't actually get treated as having stockpiled anything. In comparison, this person had 2 dispensings on the same day, and so in the output, you would actually see them only contributing 2 dispensings because the stockpiling parameter is taken into account. So this is really more for background information. It doesn't really come into play with this analysis per se. All right, so now showing you the episodes with an event. So this is just giving you a count. So these are crude data. There's no adjustment here. This is just the number of episodes that had an event of interest. And it's just a straight count. You can see the relative number for each of the different groups and for the different outcomes. So this is your overall data from the type 2 output. What you're also going to get is anything you request to be stratified. So you can ask for the data to be stratified by age, and you can specify the age groups by sex, by year, by month. It really kind of depends on what you want and what's important to you. You can also specify other strata of interest if there were conditions that you were interested in, if you want to explore effect modification or something like that. And I'll just kind of flash up the data so you get a sense of it. I think one thing to take home from this, without going into the details, is that depending on what you ask for, you end up with a lot of data. You wind up with a lot of data to review depending on how many cohorts and how many scenarios, and if you stratify by many different things, you can do it by age and sex as well. So it's just keeping in mind there's a lot that's possible. It really just depends on what you want it for. So these are the data stratified by year, and then you've got it by sex, and then you have it by age. And again, age can be specified however you like, and what you're seeing here are just the 3 age groups that were used for in the synthetic data, with this age group of overall 18 to 65. So the columns stay the same, and you're just getting more strata. So that's the type 2 output. What I'm now going to show you is some of the output from the propensity score analysis. So this is really not like propensity scores 101. I'm going to give a high-level overview, mostly from the perspective of how the tool works, but from a very high level, and then also the kind of data you're going to get back. In addition to there being obvious documentation on how the tool works on the Sentinel website, there's training materials. Jane, who's going to talk next, gave a training just on the propensity score tool a year or 2 ago. Those slides are available online, so this is really not meant to, walk away as an expert in propensity scores, or if you don't know anything about it, to necessarily have a full working understanding. But this is where we are in this chart. So it's a level 2, so we're doing causal inference analysis here. We're tacking it on to a type 2 where we're looking for an exposure and an outcome. And I'll show you now how it works in terms of the diagram. So what you can see here, we've just added this box here which lists the different factors or the covariates that were included in your propensity score model. So those are user defined. You have to decide what you want in your model. I can't remember if Judy mentioned there is a high dimensional propensity score capability as well. We're not going to talk about that here, but this is what was chosen for this analysis. So it was 1-to-1 matching. You have different calipers that you can choose. This was 0.05, age, sex, and certain conditions of interest were selected. And again, sort of at the highest level, this is what a propensity score analysis is in the CIDA tool. So you can, once you've defined your propensity score model, what you want in it, and the model runs, you're choosing whether you want to match on propensity score or you can stratify on propensity score. So what I'm showing next is the matching analysis. You'll get back Cox proportional hazards, regression, and hazard ratio, and then the 95% confidence intervals. This is just a note that you can, depending on what you're going to use the data for and the type of analysis you're going to do, there are different levels of data that are returned. Generally, as Judy mentioned, we operate under sort of minimum required standard for Sentinel, and so we try to get back as little as we can, and avoid sparse data as much as possible and avoid patient level. Sometimes certain analyses require that, so that capability is there if you needed to have patient level data. Obviously, if you're not working in a distributed data network, it's a very different kind of capability that you would have. So I'm going to show you some of the output kind of quickly. Again, it's not nearly as exciting because it's synthetic data. So Jane's going to show you the real data. But what you'll get back from the propensity score tool is you'll get your table ones, and you're going to see it unmatched, before matching and after matching. It's not exciting here. Judy already showed you this morning that these cohorts are actually quite similar, and so there's not much difference. I won't show detail, but this is unmatched analysis. And what you would do is, if you want to quickly see how balanced these two different cohorts are, is you look at the standardized difference, and anything greater than 0.1 is going to signal that there's a meaningful difference there, and you're going to want to see then, when you look in your matched output, that difference has been minimized. And if it hasn't, then you might need to do something about it, right? So we've talked about, internally, I've not been involved in analysis that did this, but you might ask for that patient level data back if you think there's a good chance that matching isn't going to get rid of that imbalance. You can then do additional adjustment after if you got the patient level data back. So that's an example where you might request that. But here, again, not very excitingly, the cohorts are quite similar and there's very little change after matching. But these are the types of data you'll get back when you run the tool. Propensity score distribution will also be returned before and after matching. Again, not particularly representative or interesting here because the groups are so well matched to begin with, essentially. So you can see that the distribution one looks a little funny. It has this hard left truncation, which is really a product of the synthetic data. But you can sort of barely see where the two groups don't overlap. And normally what we see with a propensity score distribution prior to matching is you see these two distributions with some overlap. And then after matching, the distributions are overlaid or much more closely overlaid. You're just not, it's very satisfying. You're not getting that here because of the nature of the data. And then you'll also obviously get what you're really interested in, which is your risk estimates. And so you can see here, you've got your rows of the two different cohorts, you've got your unmatched analysis on the top, and you've got one-to-one matched analysis, and you've got two different kinds of output. I won't speak about it in detail, but really, if you've done one-to-one matched analysis, you can use the unconditional output and really maximize your sample size. And that's actually the preferred approach by FDA within Sentinel. If you're doing one-to-one matching, use the unconditional so that you can take advantage of everyone in the cohort. And then I'll just quickly show you the different columns. So you've got number of users, their years at risk, the average number of days at risk, average years at risk, et cetera. And then you get into your actual risk days. So you got your incident rate, the risk per 1,000 users, incident rate difference, et cetera. And then here's your hazard ratio and 95% confidence interval. So this is your unadjusted, and then this is your one-to-one matched unconditional analysis. And you can see basically null result, and you get a p-value. So again, this will be much more exciting when Jane shows you real data. But that's the output you'll get. You'll also get kaplan-meier survival curves for time at risk. They look funny here because we've censored on end of exposure, and people had short follow-up times, short time at risk. And so it's more, again, giving you a sense of what you'll get back. And then finally, I'll show you, this is another output that you can get. This is actually not currently in the tools, but it's coming. We have data that are output, so you actually get a raw data file of attrition data. So to be able to follow where, basically where people got dropped as you created your cohort and as you conducted your analysis. It's just that right now the tool doesn't create this nice table as a report unto itself. But this will be in a future version of the tool, and it's a really useful way to make sure things make sense. It's sort of a way of quality checking your analysis and making sure everything looks okay. So that's all I have. Again, that was like a crash course in looking at the output. Hopefully it's helpful just to get oriented to it. I think we have time for a minute or two of questions. And we'll be here at the break as well as afterwards, so happy to answer any questions.
Do we have any questions? Tryptophan working? Thank you for showing us the sample output. Now we're going to actually move into 3 presentations on the case studies that actually show how the CIDA tool is used when we answer questions at FDA. To do that, we're going to bring up Jane, and she's joined Sentinel in 2016. She received her doctorate training in pharmaceutical health services research from the University of Maryland. It's right here from Maryland. So let's see. Okay, with the concentration in pharmacoepidemiology. She's also serves as a Sentinel investigator, and she actively engages in routine queries, methods, and other work groups because we put a lot of work on them, so we keep her busy. With the Sentinel leverage projects that she works with is the innovation of medical evidence development and surveillance, which is also called IMEDS. And her past queries and projects include the antipsychotic and stroke L2 pluses analyses, ICD-10 preparedness work group, and also the replication project that collaborated with the Canadian Network for Observational Drug Effect Studies, otherwise known as CNODES. Jane also contributes to various internal and external trainings that routinely lead to Sentinel's propensity score analysis training, also in the 2018 Sentinel Public Workshop. Jane?
Statics. Oops, let me just get this right. Okay, thanks, Anisha. And then good afternoon, everyone. This case study kicks off one of the three this afternoon that you're going to see, but I promise this is the last presentation that you will hear about antipsychotics today. So the case study one will present the true actual queries that we run for the antipsychotics and stroke investigation done in the Sentinel system, from feasibility assessment all the way to inferential analysis. Part of this work is accepted for publication in Journal of Clinical Psychiatry, so our special thanks to FDA members and other Sentinel Operations Center collaborators. And also many, many thanks goes to our data partners who contributed data to these queries. This case study will start with safety questions. And then I'm going to introduce how the suite of Sentinel tools provided support to various levels of analysis, from the background rate, feasibility assessment, comparative assessment, and finally briefly go over some regulatory actions taken by FDA after Sentinel results join evidence from other sources and provided for reference. Back in 2016, FDA considered a proposed label change regarding a warning or precaution against cerebrovascular events associated with antipsychotic use. As Judy already briefly went over this morning, at the time of the query of the marketed antipsychotics, only 3 of them have a label warning against such risk, and most of them, actually all of them, focus on the older adult populations and those with diagnosis of dementia. If you look into the literature, you can find that there are comparisons about the risk stroke among typical antipsychotic users, atypical antipsychotic users, or the unexposed. The available information support that there's a higher risk among typical antipsychotic users when compared to atypical antipsychotic users on their risk of stroke. However, most of the studies summarize findings from studies looking at the target population of older adults or those who has diagnosis of dementia. At that time, and given this information, FDA could not confidently label a cerebral risk or stroke warning for all age antipsychotic users. So it was unknown whether the younger or the non-demented adult populations, or even younger, the children populations, if they're on antipsychotics. These populations are often characterized with fewer or almost none similar cerebrovascular risk factors as we see in the older adults. When these group of people are on antipsychotics, do they also share the similar differential risk when they are on different types of antipsychotics? Translating the safety concerns into the study questions. Do younger, non-demented, and typical antipsychotic users have a higher risk of stroke compared to atypical antipsychotic users? Does antipsychotic dose modify this risk, particularly haloperidol? Is the risk the highest in the first few days or the first few weeks after you start antipsychotics? Do concomitant users of atypical antipsychotics and antidepressants have higher risk of stroke compared to users of the only antidepressants? Here's what happened in a typical pharmacoepidemiologic study. Researchers or investigators often go from the steps of practical research, cohort identification and characterization, statistical analysis, including confounding control, and also follow up with additional validation efforts. Over the years, Sentinel has developed a suite of programs that can support a different level of investigation. From left to right, summary table, they run the fastest, and they provide simple member count in the Sentinel distributed database as long as these members have your pre-specified diagnosis code, procedure code, or drug exposure. Level 1 analysis refer back to what Judy mentioned as CIDA type 1 through 6. They provide descriptive information and sometimes unadjusted risk estimates as well. Level 2 and Level 3 analysis, these are the inferential methods and modular programs we have as a Sentinel tools. They include the self-control risk interval, propensity score analysis, and also the prospective sequential surveillance modules. The follow-up and sensitivity analysis available under Sentinel tool are those that can be done using the frozen data. For example, PEPPER, which is the patient episode profile retrieval. They are essentially the line list of all claims of the patients you identify with a certain exposure or a certain outcome. It's a very useful step before you go ahead and do the actual chart extraction from the data partners or single data source to conduct the chart review. The usefulness of these PEPPER records will depend on the exposure of interest or outcome of interest. For the antipsychotic query, we knew at the time the antipsychotics were prevalent, especially in the older adults, for both indicated or off-label use. But we were not sure how many of the younger adults in the Sentinel population that were on or used any of the atypical or typical antipsychotics. So we went ahead to run a summary table query, which provides the fastest and preliminary counts of members on these drugs. So this is a screenshot from the Sentinel website. Summary table are part of the routine query tools, and then you can find all associated documentation on our website. Essentially, they are pre-populated tables. Therefore, that's the reason why they can provide simple and rapid menu-driven member counts in the database. Here, we provide 8 prevalence tables and 3 incidence tables looking at members with pre-specified diagnosis, procedures, or drug exposures. When they are incident tables, you can optionally apply 90-day, 183 days, or 2,070 days washout, specifying these, for example, drug users without previous exposure. There are 2 ways to identify members exposed to certain drugs. One, you identify or define the drug exposure using generic names. Alternatively, you can also identify the drug exposure using drug classes. For the antipsychotic query, we chose the method I here, which is identify the users of antipsychotic using their generic name. Highlight or example output from summary table. These tables, we usually return results in pivot table format. For example, what you see on the screen here, these refer to how many users that we found in the database that has evidence of prevalent olanzapine use. By default, we have the stratification by calendar year, sex, and pre-specified age groups. You can change this age group cutoffs in the way when you enter the parameter modules. And then for the prevalent tables, you can see the number of users for these drugs, number of dispensings, and total day supply based on these dispensing information. From these results, we concluded that for each table of the antipsychotic that we chose, we still observe quite significant amount of the younger antipsychotic users aged 65 or below, actually 64 or below. So FDA made this decision that we have probably enough sample size to move on to level 1 analysis. So level 1 analysis, remember that it's mostly for feasibility assessment. In our example here, we use CIDA type 2 analysis type 2 analysis to establish new user cohorts of antipsychotics. In the design diagram, they very much resembles what Judy showed and Noel showed in the mock queries. Essentially, if you are atypical antipsychotic new user, we define an index state based on the day that you have a new dispensing of atypical antipsychotics. And then to qualify or be eligible for a new user, you need to have no typical or atypical antipsychotic use in the 183 days prior. During the same evaluation window, we require members to have continuous enrollment 183 days in the valid health insurance or drug insurance plans. We also exclude members who had recent stroke in the 60 days prior to the index date. In the post-index window, we follow these members up on their exposed time, up until to the earliest of either event outcome occurrence, in our case that would be stroke, or the earliest of the censoring criteria met. It could be a natural end of an exposed episode, or the initiation of the comparator treatment. Query end date, data end date, anytime the member is disenrolled from the single insurance plan, or recorded death. For this query, we also were interested to learn about the cohort baseline characteristic once each exposure cohort were defined. So for example, it was one of the study questions to look at how many members of our cohort will simultaneously have on baseline SSRIs at least 60 days prior to the index date. We were also interested in the chronic conditions in the past 6 months prior to the index date for the cohort members. So remember that level 1 analysis, they are designed to prep you better or inform your level 2 analysis decisions. You are able to vary the definitions of your cohort. However, in level 1 analysis, once you change the definitions of exposure, incidence, enrollment time, or outcome requirement, or even inclusion and exclusion, you will need to create a different scenario in level 1 analysis in order to see these changes and their impact on sample size. The covariate provides a more flexible approach to quickly examine roughly how many people you will have sample gain or sample loss once you apply a longer inclusion and exclusion criteria after your level 1 analysis. I will elaborate this a little bit more in the next slide. The bottom line is that when you change the core definition involving exposure and outcome, you will need to create different scenarios to see the changes. Okay. And last but not least, CIDA also provides the functionality of code breakdown, looking at your index event, be it a diagnosis, procedure, or drug exposure, or providing the same for the outcome breakdown. For example, if you are interested to know how many atypical antipsychotic new users started their antipsychotic with clozapine versus olanzapine, this functionality will help you identify the proportion of members. And then similarly, you can also identify for those antipsychotic users who experience outcome at the end of the study, how many of them actually suffers from ischemic stroke versus intracranial hemorrhage. Highlight of level 1 results. Unfortunately, given the limitation of time, I will not be able to show the full results. But some useful information, like Noel already mentioned, we provide the baseline characteristic table, or AKA table 1s, for each of the exposure cohort that we defined in our level 1 analysis. For the 3 columns shows here, you have one cohort, newly initiated atypical antipsychotics. Another column, another cohort, or another scenario that defined those members who newly initiated typical antipsychotics. And a third one who newly initiated the haloperidol. We identify the age on index date information, also provide other demographic information such as sex and race and certain data partners. And remember that we had one of the study questions that are particularly interested to see whether dose modify the stroke risk among different antipsychotic users. And then particularly when we look at haloperidol, we leverage the covariate function to explore the option whether we can proxy this daily dose question using the index product strengths. We had this trial because the product strengths instead of daily dose are readily available in Sentinel data model. And most of the patients do not change their haloperidol dose in a short period of time, at least when they newly started haloperidol. So we were trying this option, but we found out that most of the patients, if they newly initiated haloperidol, they actually over 2/3 of them are on the low dose side. So if we do the level 2 analysis, which is a comparative assessment stratified by the haloperidol dose, we may experience some sample size issue. So this is one example how you use the level 1 results or finding to inform your final decision in the comparative assessment. Another highlight on this table is that remember that we exclude members who ever had a recent stroke. You can also try to identify among these people how many of them additionally have stroke outside of the recent definition, which was 60 days prior to index date. So we can use stroke in prior 3 to 6 months or negative 61 days all the way back to negative 183 days. This gives you a rough sense of up to how many antipsychotic users with stroke history we lose if we extend the stroke exclusion all the way from 2 to 6 months prior to the index date. Similarly, you can do the same for the SSRI baseline use. This gives us the information up to how many concomitant SSRI users we gain if we extend the concomitancy definition from 2 to 6 months prior to index date. So these are the informations that you can try to gauge the sample size change when you make final design decisions for level 2 analysis. This is a very abbreviated version of what Noel just showed in the type 1 output. For each exposure cohort, you will see the number of total users and time at risk. And then we have separate columns that can provide information for you to derive incidence or incidence rate if they're not already there. So once we check the feasibility in the database, we have enough number of antipsychotic users on each class of antipsychotic. FDA decided to move forward to level 2 propensity score analysis. We call our study questions, we can actually group them into 2 aims. The first aim focuses on typical versus atypical antipsychotics without any consideration of concomitant SSRI use. And based on L1 results, we knew that Sentinel might not be the best source to tackle the dose problem. And then in the differential analysis, we actually removed this question and focus on the other 2. The rest of the aim 1 here were relatively straightforward. This is a screenshot of a query specification, a document we prepare for every query when we assemble the package. You can see that we established 4 different comparisons. The first comparison compare all typical antipsychotic users versus any of the atypical antipsychotic users. The middle 2 columns specify different risk windows during follow-up to identify event. The last column group on the right compare haloperidol users only to any atypical antipsychotic users. And then for all comparisons, we apply the age restriction, 18 to 64 years old, or the dementia exclusion. Now that was aim 1. For aim 2, this question has concomitant users of 2 different drug classes, atypical antipsychotics and antidepressants versus antidepressant users on their risk of stroke. Essentially, you're comparing the co-medication people versus the monotherapy people. And then in the context of comparative assessment, who are we actually comparing the antipsychotic user to? That is a challenging question because CIDA only works in a way that you need to find the index event for either side of the comparison treatment group. We came up with 4 options for the comparator for this AIM2. Antipsychotic users themselves, or the non-users, antidepressant users, or the negative controls. For option 1, antipsychotic users themselves, this involves self-control design. At the time of the query, Sentinel tools only have the self-control risk interval readily available as a modular program functions. And then there's a very important assumption in the self-control risk interval design is that the outcome should not change your probability of getting either treatment exposures in your study. And that is not the case for antipsychotic and stroke. Because remember, we already know that there are 3 stroke warnings on the atypical antipsychotics in particular. So we move on to option 2, the non-users. We consider exact match on age, sex, and or calendar time. Exact match is now a function or capability within CIDA, but it was not available back in 2016 when FDA brought this question to us. At the time, if we were to do the exact match in place of propensity score matching, it would require a significant amount of custom programming attached to the existing CIDA package or modular programs. So the whole time and effort would go beyond the scope of Sentinel queries. So we did not use this option 2 either. Option 3, antidepressant users. This involves prevalent new user design. And again, the query itself actually predates any detailed information that you can find on the prevalent new user design or publication. And then CIDA, for CIDA to accommodate the index state selection mechanism recommended by the authors of prevalent new user design, it will take also significant amount of custom programming work to put them together and make it automated. So option 3 was not our to-go choice at the time either. That left us option 4, negative controls, or identify a group of users of another drug class with similar indications but no known associated stroke risk. There's some back and forth between us and FDA members, and then we determined that Z hypnotics can be the appropriate drug class to serve as our negative controls. So in the end, for this specific AIM2, the final comparison was actually the antipsychotic users, atypical antipsychotic users compared to the hypnotic users, and both groups with baseline SSRI use. For both aims, the first aim comparing different class of antipsychotics, and the second aim comparing antipsychotics versus the hypnotics, we use a one-to-one propensity score matching method. Propensity score summarizes a number of member characteristics into a single variable, which then reduce the need to put a lot of covariates into your outcome model or outcome analysis. Essentially, we fit a logistic regression, and then based on baseline characteristic, we calculate for each member their probability of receiving the exposure treatment. In our case, that would be the typical antipsychotics. Once every member has a propensity score calculated, we match them one-to-one based on their propensity score. Sentinel tool also has a function, an option, that you can do variable ratio matching up to 1 to 10. And then all the matching is performed by nearest neighbor approach. For more information, you're welcome to refer to our last year's training. There's a full hour or at least 45 minutes training talking about the propensity score analysis tool under Sentinel. A quick highlight of level 2 results. This is looking at the first aim, where we only compare typical antipsychotics versus atypical antipsychotics. As Noel mentioned, we all by default provide the table 1 characteristic. What's a little bit different is that in the level 2 analysis, we provide one table 1 for the unmatched cohort and the other table 1 for the matched cohort. And then you can see the total number of people in each cohort by exposure, baseline demographic, and then chronic conditions we specified to have or not have in the past 6 months prior to index date. Between the unmatched and the matched cohort, you can see for antipsychotic query, we lost a significant amount of people after propensity score matching. We actually lost over 90% of atypical antipsychotic users. This is a very classic bias efficiency trade-off. And especially in the case that we know there are potentially many, many more off-label users of atypical antipsychotics compared to typical antipsychotics. The decision was to move forward with propensity score matching and then use the risk estimates in the unmatched cohort and the matched cohort. And Noel already mentioned, we also provide balanced diagnosis using standardized differences. Okay. The other level 2 results involve the risk estimates. By default, the propensity score analysis tool used the time to event analysis based on Cox proportional hazard model. The hazard ratios, if you look at the results in the unmatched side, we observe elevated risk for the elevated risk of stroke for the overall analysis in the first 15-day analysis. They have a higher risk of stroke among the typical antipsychotic users compared to atypical antipsychotic users. After propensity score matching in the matched cohort, however, hazard ratios all cross the null, regardless the overall analysis or risk window specific analysis or the haloperidol analysis. The tool also provides a KM survival curve by default. You are able to observe the different survival curve by exposure group, and then the number of people remaining in each exposure group after different time points at the bottom. You can see some differences in the output for the first probably 2 weeks, but if you look closer, the absolute difference in survival probability is small on the scale. Moving forward to the AIM-2 results. This is a slightly different comparison. Remember that we end up choosing the atypical antipsychotics and comparing them with Z-hypnotic users. Both of them has baseline SSRI use. Hazard ratio suggests none of the hazard ratios actually in the unmatched cohort suggest any significant elevated risk. And then after propensity score matching in the matched cohort, all hazard ratios cross the null, supporting that none of the comparisons actually shows elevated risk of stroke between the atypical antipsychotic users versus the Z-hypnotic users, even though there is a change in the effect estimates for the overall study. Some conclusions and discussion. There were no significant associations found in either study. AIM-1 compared the typical antipsychotics versus atypical antipsychotic users. We observed crude increased hazard ratios adjusted away after propensity score matching. AIM-2 compared atypical antipsychotic users versus Z-hypnotic users. We observed modestly but non-significant increased hazard ratios. And then, based on the confidence intervals we observe, we cannot completely rule out increased risk for the comparisons we conducted. Overall, the event rate of stroke are low in the younger non-demented population. And then propensity score matching, this is actually one of our limitations. It reduced largely the sample size before and after matching. It sacrificed some of the precision of our estimates. However, it's a good trade-off, in our opinion, with improved confounding adjustment. We were not looking at the subgroup risk by age group or dose in these analyses, but there are good future steps if there are further risk stratified by these risk factors that can be identified in the literature. It could be an upcoming query next. Using the Sentinel results along with evidence from other data sources available to FDA, in the end, the final decision was no label change necessary for the antipsychotic users. And the query results were presented at the 2017 International Conference for Pharmacoepidemiology and Therapeutic Risk Management. Part of this work is accepted for publication in Journal of Clinical Psychiatry. Every query report and also the ICP presentation we posted online on Sentinel website. It's freely downloadable. Also, for more information, we post AIM-1 and AIM-2 specifications, reports, or the relevant code list online. You are welcome to click over the link or search and find out about these details. And as Judy mentioned, this evening, we also uploaded the entire analytic packages about these two aims available to download from our website. That's all for case study 1, and I'm happy to take any questions you may have.
All right, so case study 2, what comes after this is coffee. So we're close. I'm going to be presenting on a set of analyses that were conducted within the Sentinel system last year, and it's on biosimilars. So I am going to be talking obviously about biosimilars, but really the intent of this, like our other discussions so far today, are to give you a sense of the capabilities within the Sentinel analytic tools and to really give you that perspective less than the results themselves in this case. I'll give you enough background to be able to follow along in terms of what biosimilars are and why they're of interest to FDA in this setting, and to be able to interpret the results. But it's not going to be a lot of detail on biosimilar products themselves. So first off, I'd like to thank everyone who is involved in this work. I'm actually presenting on their behalf. I was not involved in the analyses themselves. So Sarah, Effie, and Michael at FDA, Kathy, Elnara, and Libby at the Operations Center, and then all the data partners who participated in the analyses as well. So to start off, again, I am going to give you the highest level overview of what a biologic is, what a biosimilar is, how they're regulated as a non-regulator. So I'm giving you enough to be able to follow along and understand the intent of the questions and how these analyses were run and what the purpose was. So the highest level biologic is a product that is generally derived from living organisms. It includes vaccines, monoclonal antibodies, antitoxins, all those kinds of products. They're obviously manufactured in a very different way than typical small molecule drugs. So not only are they much smaller, so you can see in this image, there's a little pull-out box that's showing you the relative size and complexity of aspirin compared to the, kind of looks like a monster of monoclonal antibody in comparison. But basically the point here is that biologics are much bigger, they're much more complex, they're manufactured differently, they're also regulated differently. And so with all of that as part of the setup, I'll go into a little bit more of the background. The other thing to note is that biologics, again, is sort of the highest level of understanding the regulatory processes. They're approved in a standalone development kind of way. So there's preclinical and clinical data that go along with each product, and they go through the sort of typical channels. It's quite different for biosimilars. So biosimilars, so if you think of the biologic as the reference product, the biosimilar is being compared to that reference product. And conceptually, it's like the difference between a brand and a generic on a high level. So a biosimilar is a product that is highly similar to the reference product and is expected to have no differences that are clinically meaningful. So the expectation and the demonstration of evidence must show that in order to be considered a biosimilar, these products are virtually the same in terms of safety, purity, and potency of the product. The note here about interchangeability, so brands and generic typical drugs, those are considered interchangeable. So if you get a prescription for a brand, a pharmacist can interchange that prescription with a generic instead. It happens on that level. That expectation and allowance of interchangeability does not exist necessarily for biologics and biosimilars. So this is, again, the framework of the brand and the generic is useful, but it doesn't exactly follow through for biologics and biosimilars. The expectation is that there's no clinically meaningful result or no clinically meaningful differences between the products. We expect that basically they're going to behave the same way. And there's this abbreviated approval pathway that's been established by FDA for approval of biosimilars. So I won't go into the history of the legislation underlying this, but the point here is that for biosimilars, for these products, they basically get approved in an abbreviated way, and they're actually they're not coming with their own dossier of preclinical and clinical data. It's an extrapolation of the biologic product to the biosimilar once that biosimilarity is demonstrated. So there's an extrapolation also to the indication. So as you'll see, if the reference product, if that biologic is indicated for 5 conditions, a biosimilar to that product will also be approved for those conditions. The biosimilar does not have to independently demonstrate all of the safety and effectiveness data. So that's the context of what biosimilars are and how they're approved. So why is FDA interested in using a system like Sentinel, so interested in doing post-market surveillance of these products? So biologics, biosimilars are complex. They have inherent variability just by the nature of what they are. While there is an expectation of some lot-to-lot variation, the intent is that they don't vary enough to have a clinically meaningful difference. And so the other thing to note is that immunogenicity and related adverse events is a concern for biologics in general. And so the expectation is that those kinds of adverse events would not differ between the reference product, the biologic, and its biosimilar. So FDA wants to be able to establish that and monitor these products in real time, in real world data, in order to be able to ensure their safety. The other question to this is, why now? Why was FDA interested in studying these last year and currently? And it's really, as you'll see in a second, because there's more and more of biosimilars on the market. So this is as of the end of 2018. You can see the currently approved biosimilars in the United States. So I'll orient you to the table. So you have the reference biologic here by proprietary name. In the middle, you have the biosimilars associated with them, their proprietary name and their non-proprietary name, along with a 4-letter suffix, and then also the biosimilar approval date. So a few things to note here. First, some of these products, particularly the ones that were approved more recently, they're not even necessarily marketed yet. The first product up here, the first biosimilar, filgrastim, is actually not technically a biosimilar because it was approved prior to the establishment of the biosimilar approval pathway by FDA. So you can see these four-letter suffixes at the end of each of the non-proprietary names of the biosimilars. Those are to be able to identify the individual product for pharmacovigilance purposes. What you're seeing here circled in red are the products we're going to talk about. These are the ones that FDA examined in the Sentinel queries. So Neupogen is the proprietary name for the reference biologic, and that's for filgrastim. And then you can see the biosimilars that were approved at the time and had data established for them for that product, so Granix and Zarzio. So filgrastim is used to treat neutropenia, so low white blood counts in patients who are being treated for cancer to avoid and prevent infection. Remicade is the reference biologic for infliximab. Infliximab is indicated for 7 different autoimmune conditions, such as ulcerative colitis and Crohn's disease. And you can see the 2 biosimilars that were approved with some data available at the time that the analyses were run, so included in these analyses that I'm about to show. So like everything in claims data or any healthcare data, everything is about how are you going to define your exposures or outcomes of interest, what codes are you going to use. And so for biosimilars, as I said, there's the proprietary name and the non-proprietary name and the 4-letter suffix associated with it. That's great. How are we going to actually capture these in the Sentinel data? So what's available for these kinds of products in the Sentinel Common Data Model, in claims data in general, are procedure codes and drug codes. So specifically the procedure code system that is used for these kinds of products is called, for short, HCPCS, Healthcare Common Procedure Coding System. And these are for products that are administered in a healthcare setting. So these drugs are administered by injection or infusion. So if I go in and I receive one of these, you would expect to see, likely, one of these procedure codes because I've received it in a doctor's office and they want to get paid for that. If you are actually dispensed one of the drugs to administer at home, it would show up in our typical outpatient pharmacy data, and those are captured with NDCs. So there are 2 different ways that we assume we would see these products in claims data. And you might see both for the same treatment. So perhaps I've gone in and for some reason you might see both a procedure code and an NDC for the same actual treatment. This is the set of questions that FDA had coming into this analysis. So overall, the purpose was to be able to describe patients who are exposed to biologics and biosimilars in the Sentinel distributed database in the Common Data Model. So how are they identified and captured? So that's the overarching sort of descriptive exploratory approach. And then there were these specific questions that FDA wanted to answer. So first, how are these products captured? Are they identified by HCPCS codes, by drug codes, so in other words, administrations in a healthcare setting or an outpatient pharmacy dispensing, or both? How did the data look in terms of time trends? As you'll see, there was a published report on one of the set of products, and the question was, can we see similar trends in Sentinel? How did the data look? Third, are there potentially missing patients? And you'll see in a moment, there are these non-specific codes that can be used for biologics and for drugs. And so the question was, are we going to see those kinds of non-specific codes in the system, which would be problematic? The next question is getting at this issue of the extrapolation of indications. So specifically for infliximab, it is, there are 7 indications associated with infliximab, and so the question was, if you compare the reference product to the biosimilars, how does do the patient characteristics differ, or are they the same? And then last, there was use of the tool that Judy demonstrated this morning, so the medical product utilization tool, which has this ability to give you all sorts of information on patterns of use. And so the question was, basically, what do we see in terms of timing of these codes? How often is someone receiving one of these products? How do the data look in Sentinel? And so what I'll do is I'm going to walk through how each one of these questions was addressed, what the study, the design diagram looks like, and then some of the data. So going with the first one, so how are these products captured in claims data in the Common Data Model? What are we going to see in terms of procedure codes / administrations or outpatient pharmacy dispensings? So you can see here a very simplistic study design diagram. And you can see here, sort of it's you know, going for everyone. Find us everyone who has one of these products in their history in the query period. So I'll just walk you through this. So in order to be included in this analysis, you just had to have 1 day of either of medical and drug coverage during the query period, basically. So there was no prior enrollment requirement. We didn't say you have to be enrolled 6 months or a year, because we weren't looking for history of anything. This is just count the number of procedure codes, count the number of drug codes. So there was no prior enrollment requirement. There was no washout. We weren't looking for incident use of these products. And it wasn't in a particular patient population. So we didn't say kick out anyone who has dementia or only include people with dementia. So it was very simplistic. And we said, look for all of the valid index dates. So this is different than the query we've been talking about the rest of the day, where we only took the first of your antipsychotic. This is saying, every time I go in and I get a dispensing or a procedure for one of these drugs, count all of them, because I just want this global perspective of how much use is there. And we did it separately for HCPCS and procedure codes. So again, you basically just had to be in the system with at least 1 day of medical and drug coverage to be one of those eligible members. Okay, so this is showing you the codes that were used in these queries. So you've got your non-proprietary name and the proprietary name, the HCPCS codes that were available, and the NDC. So I'm not going to belabor this, but for a period of time, and it actually ended in early 2018, so the HCPCS had this 2-digit modifier, which actually is not in the Sentinel Common Data Model, so we couldn't capture those. But it's just a side note and doesn't, we're not going to dwell on too much. But the other thing to notice here is these two categories, or these two codes that are just non-specific. So a drug or a biologic. And the question was, are we going to see a lot of the use of these drugs in relation to these products? And that would be a concern for FDA. So getting right into the data. So this is the output for filgrastim, right? So this is, again, the product that's used for neutropenia. And so you can see here, you've got Neupogen, which is the reference product, and then the 2 biosimilars. And there's the data by procedure code, so administrations, or the data by dispensings, NDCs. This is your outpatient pharmacy dispensings. These two groups of data are not mutually exclusive. So if I got both a HCPCS code, a procedure, and an NDC, I'm counted in both of these categories. And you can see the relative number of Neupogen in comparison to the biosimilar. So it's not surprising Neupogen was on the market a lot longer. There's more use of it. There's less use of the other 2 products. You can also see the mean number of codes per patient. So there's a little bit less per patient for the biosimilars than the reference product, and that might just be a function of time. It might be actual use, but it's giving you a window into what's going on. And so I don't think I mentioned this out loud, but it was shown on the screen. So the query period for this was going from early 2015 into 2018, but actually most of the data partners had data into like late 2017. And you'll see the consequence of that sort of incomplete data at the end when I show you some of the time trends. But this is basically showing you that both types of codes are used, so both the procedure codes and the drug codes, but there's much more use of the procedure codes. So this is getting at that second question that FDA had, which is, what do the time trends look like? Do we see an uptake that's been reported elsewhere? So this is the data that was reported in a paper in JAMA showing the use of filgrastim. So this is the reference product, Neupogen. You can see its use over time declines as the biosimilars increase. And so you can see the 2 biosimilars here, each one individually, and then you can see the 2 biosimilars combined. And I would just note the time frame here is going from 2014 to the end of 2016. So it's different than our query period. But the question was, for FDA, do we see similar trends like this? We would expect to. The other thing to note is that this is the Medicare population. So oftentimes there will be something that was done in Medicare, and the question is, how does it look when we run it in Sentinel, either among that same older age group or just in general, or maybe just among younger adults as well. So that's what they were aiming to replicate. And this is what came out of Sentinel. So this is looking at just the administration. So this is just the HCPCS codes over time. And you can see that the originator product, the reference product, its use is declining over time as the use of the biosimilars increases. A couple things to note here. So one, I told you we don't have data through the end of 2017 for all the data partners, so that's why things drop off. It's a function of the data. The other thing is, I think in our world, everything really rides on how codes are used and hoping that they're used exactly as intended. And you can see actually here they are. So the green line is the code that was used for Granix for some time, and then there was a code switch right in January of 2016, and it shows up perfectly. I don't know, it's very satisfying to see something simple like this show up well. So that's just showing you that there was a code switch and we see it and everyone used the appropriate code afterwards. It's similar to what we see at the ICD-9, 10 switch when we're looking at diagnosis codes over time. You want to see that exact moment, and generally it does correspond that way. So the point being that the time trends that were observed in that paper that was reporting on Medicare use, comparable results were seen in Sentinel for a different or slightly different time period, but reassuring that the trends were basically the same. So going back to the first question of what kind of codes are being used and what are we seeing in Sentinel for infliximab, for its biologic reference product and its biosimilars. So a few things to note here. So Remicade is the reference product, was on the market for longer, so there's much more use. And these biosimilars were quite recently approved in reference to the query period. So Renflexis actually was approved in early 2017, so there was really very little use at the time that this query was run. So you correspondingly see low use of the biosimilar. And Inflectra was approved in April 2016. Take home here, similar to filgrastim, is that you're seeing use of both kinds of codes, but more dramatically here, you're seeing much more of the HCPCS of the procedure administrations versus the drug codes for infliximab. So again, infliximab is the set of products that's used for autoimmune conditions such as ulcerative colitis. So now I will move on to that exploration of, do we think we're going to miss a lot of people? Are people getting these products and those specific product codes are not being used? So this was an exploration of those unclassified codes. So the way that the study team set this up was they said, find everyone who has a dispensing for one of the products, so one of the NDCs, and then look in the 3 days before and the 3 days after for one of the unclassified biologics or unclassified drug codes and see what we see. So it's not telling you a couple things. It's not telling you for sure these are actually associated with those products, because maybe it's representing a totally different product. And it's not telling you whether they're being used at other times, but it was a proxy to get a sense of, is there a lot of use of these non-specific codes, which could be problematic? And the short answer is there doesn't seem to be much, at least in the way that this analysis was set up. So again, I had a dispensing of Neupogen, and then there was looked in a window of the 3 days before and 3 days after that dispensing to see if I had an unclassified biologic code or an unclassified drug code, and it's pretty low for the specific codes. And then, so this is for filgrastim, and you can see the same thing for infliximab with even less use. So exploratory work just to get a sense of, are these non-specific codes showing up, and at least in the way that that query was set up, it suggests that they're not being used very much, which is good. So now moving on to the next question that FDA has. So this is the fourth question. It's asking about, how do the patient characteristics differ, or are they similar when you compare the reference product and the biosimilar? So the setup here is a little bit, is slightly more complex, but still pretty simplistic. We looked for people who had at least a year of medical and drug coverage, right? Because now we want to see, do you have history of the conditions that these drugs are indicated for? And so we wanted to look back 1 year among people who had coverage of both kinds. We didn't have a washout for infliximab. We didn't care if it was incident use or not. No inclusion-exclusion criteria, so we took everybody regardless of what their history was. And then we looked for the indication of interest in the 1 year prior. For the reference product, both HCPCS and NDCs were assessed, so if you had one or the other, you were counted in the cohort. And for the biosimilars, only the NDCs were used because they were more specific than the HCPCS at the time that this analysis was run. So this is part of the data. So this is like that table 1 that you saw before. So it's that capability that you can get out of CIDA to get information on your cohorts of interest. So a couple things to point out here. So this is, again, infliximab. This is your reference product. These are your 2 biosimilars. One thing to note, for those of you who are paying attention, there was about 77,000 Remicade users in one of the prior slides. There's less here because we had that 1 year of enrollment criteria, 1 year of enrollment requirement. So as soon as you say you have to be enrolled at least 1 year, you're going to lose some people by having that requirement. You can see very little use of Inflectra, and then no use of Renflexis in this analysis. But you can also see obviously what was most interest to FDA was how the patient cohorts are similar or different. So the Inflectra users are a little bit older, so their mean age was 59 compared to 52. There were more females. You can see here all of the Inflectra users were in 2017, of course, because that's when they were approved. So sometimes, we'll use these are like good data sometimes just for us to check when we've got like a bunch of products and a bunch of code lists and we're wanting to make sure things make sense. You always want to, okay, we're seeing none in 2016. When was the drug approved again? So this is some of the sort of high level quality control kind of review we'll do. But then getting at what FDA was really interested, which was, what's in the history of these users' claims data in terms of their conditions. So for the GI conditions, so for the gastrointestinal conditions that these products are approved for, which is ulcerative colitis and Crohn's disease, about half of the reference product users had history of one of those conditions compared to less than 14% in the biosimilars. Again, paying attention to the small numbers. So this is, early exploratory work. Similarly, excuse me, the non-GI conditions, which include rheumatoid arthritis and psoriasis, you can see again, about half of people have one of those conditions in the 1 year prior to their use of these products, compared to 80%. So many more of the infliximab biosimilar users have non-GI conditions, and more have neither of those conditions in their history. So about 9% don't have any of those conditions in the 1 year prior. Again, keeping in mind the small number, relatively speaking, for the biosimilar users. All right, so for the last set of data that I'm going to show, this is the last question FDA had, which was around basically patterns of use. How often are people using these drugs? How often are they getting administrations or dispensings? And so this is that type 5 tool. This is the medical utilization tool. This is also the one that's built into that query builder that Judy demonstrated this morning. This is just one of the set of data, one of the capabilities within that tool. It's actually, when we run one of those analyses, there's a ton of data that comes back from it. This is just I'm just gonna show you information on the gaps between the first administration and the second. But not only will you get information back on all of the gaps, but you'll get information on days supplied and a bunch of other metrics that are of use, but in this context, we're just focusing on the gaps. So in terms of the setup, it's very similar to the first analysis where very few cohort entry criteria, only had to have 1 day of medical and drug coverage during the query period, and this is looking at just the procedure codes, so just the administrations. And we're going to get information back on the time between the first one and the second one among people who had at least 2. And this, I mean, to give you the context for this, so this will help inform in future analyses if FDA has to make decisions about what, understanding length of episodes and time on drug. This sort of background information will help them understand that as they design future queries. So that's the reason to do this kind of analysis. Okay, so this is information on filgrastim. So this is showing you the total number of administrations. So for Neupogen, for the reference product, there's more than 134,000. There were among them, so there are nearly 19,000 patients who had 2 or more administrations of that drug. The median time between the first and the second is 12 days. You get the interquartile range and the mean, and then you get the same information for the 2 biosimilars. So, as we saw before, much less use, relatively speaking, of the biosimilars, but generally the trends are similar in terms of the median time between the first and second administration of these drugs, and the mean, it's a little bit shorter here. It generally agrees with what's written in the label in terms of how this drug is used. So again, this is background information to help inform future studies. So moving on to infliximab, so similar setup, same arrangement of the data. You can see that more than 500,000 administrations of the reference product, 64,000 patients had at least 2. There's a much longer time, median time between the first and second. And you can see the biosimilar here for infliximab. There's less use, but pretty similar in terms of median and mean time between the first and second. So again, this is really background information to inform perhaps type 2 analysis. So if there was going to be an investigation of an exposure and an outcome to inform the windows of time where you might look for time at risk. So in summary, the first few bullet points are really about the results themselves with respect to the biologic products. So both filgrastim and infliximab are billed generally more as procedure codes, as administrations, but we also see a fair amount of NDCs, particularly for filgrastim. At least this preliminary assessment, to the best of our knowledge that we can understand it, the unclassified codes seem to not be used very much. The data on observed gaps, so that time between the first and second, aligns with what's written in the label in terms of how the drug should be used. So that's about the data, but that's not, yes, that's what this analysis was about, but in terms of the presentation today and what's useful for you is I think it gives you another sense of how the Sentinel Common Data Model, the distributed network, and the tools can be used to explore different kinds of products. And then, again, highlighting the range of capabilities that are available within CIDA and its suite of analytic tools. So that's it for the second case study. Any questions?
Oh boy, it's too high. Thank you for your presentation. I just have 2 clarifying questions.
Sure.
I'm Carla from Kaiser Permanente, but I'm transitioning to IMED. So
got it. Okay.
I just wanted to ask: are you limited to covariate? Is one limited to covariate assessment through just the baseline period, or can you also assess after index date?
You can assess after index date. Yeah. So if you want to, in some situations, you may want to look just forward or back and forward. So you can do both. You can basically set up a negative or positive range of numbers.
And I know you had it there that, can you define disenrollment in the observation time, not in the baseline period, as also like allowing for gaps in time? Or does it have to be like 1 day?
Oh, I'm not sure about that. I might look to any of my colleagues. So if there's no, I think if it says you're disenrolled, if there's a disenrollment date in your data, then you are disenrolled. I think that's just the way. That's the way it works. So period. That's
usually, I mean, in the system that I'm used to, there's a date that's an end date or a disenrollment date, but then for the same reasons as in the enrollment period,
you look a little bit further out.
Yeah.
Yeah.
Might be
I don't think our tools do that.
Okay.
I'm looking to. Here we go. That's why there's multiple of us.
I want to add a response. So enrollment is processed at a higher level before you even put or specify any cohort.
Okay,
right. Remember, there's probably a parameter that Judy mentioned. There's an enrollment gap of 45 days. That's our default, 1.5 months. So that's string up all the enrollment episodes before you even identify exposed cohort. So for disenrollment anytime, as long as they're greater than 45 days, for example, they will become separate enrollment periods.
Okay, that answers my question. Yes. Thank you. And then, I'm sorry, forgive me if I missed it, but did you go over, this is all the regression analyses you presented in studies 1 and 2, the case studies, are distributed, right? So the site adjustment is just the propensity score adjustment within each site?
The propensity scores are calculated within each site.
Yeah.
Yep.
And the method of
how you get to a final
distributed regression, is that sum of squares, or what do you use to?
So it's like a meta-analysis. So we get back the site-specific data, and then it's like a meta-analysis that's calculated to get the overall. There's also a distributed regression capability. We don't have a tool for that.
Okay.
Yeah.
So you use standard kind of like meta-analysis methods to do the Cox regression. Okay, thank you.
All right. Well, I'm going to hand it over to Anissa if there's any housekeeping.
One question. I was wondering about the Frederick Nyberg AstraZeneca. I was wondering about the practical details because it was mentioned before that if you want to do sensitivity, you want to alter something, do sensitivity. Basically, you need to define a new analysis.
Yep.
But on the other hand, if it's a small change, basically lots of the calculations are just being duplicated. So how is the coding actually done? So if you send out 20 different analyses and they have very minor differences, does it still take 20 times the time for them to run?
I'm looking
so is that something you have to think about when you send out your request?
Yeah,
so you can't send out 500 analyses, for example?
Yeah, so I'll give you the high level understanding as a just an epidemiologist understands it. So we generally have some rule of thumb around in terms of one query going to data partners, essentially how big it can be in terms of the number of scenarios. So we try not to have 100 scenarios. So there is actually now within our group, some of the analysts we work with can calculate the complexity of the analysis that takes into account a number of things to get a sense of how big is it. The other thing to know is that, I don't think Judy mentioned it earlier, the sort of the flow of how this work is done. So at the high level, FDA says we want to ask this question, we work with them to specify it and design it. And then before we send out a program to the 17 data partners, we actually run it internally on market scan data that looks like the Sentinel Common Data Model. And that is a good indicator of how big is the program. So if it takes 5 days to run on that, and sometimes it does, and sometimes we still send it out. Sometimes some of our packages are quite big, even if they're analytically simple. It really depends on what's in the analysis. So that's the degree that I understand as an epidemiologist. There are other people who can answer it on a more technical level in terms of what makes it take longer or shorter. So Tiffany in the back can answer that question either now or at the break.
Okay.
Yeah.
Thank you.
All right, I think we're getting closer and closer. I know that some folks may phase in and out. If you decide that this is the end and you're not going to come back after the break, if you don't mind, please do 2 things for me. One, if you take your name badge off, there's a small, Tiffany actually had, thank you, Tiffany. She has a clear holder, so if you can put those in there, we would like to recycle those. The second thing, if you, again, obviously use your app, at the very end, there's a closing session that's going to take place at 3:30. If you click inside of there and you go to the bottom, there's a post-training evaluation survey. We really appreciate it if before you leave, you can just kind of pop in there and go through the survey, because everybody's working really hard and we just want to showcase all of their hard work and see how it's impacting you guys. The kiosk is still open because I think a few of us might, including myself, may need a soda or something like that, just to make sure you do that, because they'll be closing after this break. But we do have a little bit of cushion. We won't come back until 3 o'clock. So instead of the 15 minutes, we actually have 20. All right. Thank you so much. And those online, we appreciate you guys hanging in there as well.